All right, sorry for being a few minutes late. Kind of hard to get my ass out of my chair, basically, when I was eating my lunch. Okay. Hello. And my other cat coming back, ready to take up her usual position. All right, so get myself comfy. Essentially, what we're going to be doing today is, yes, all right, sure. She's trying to make me pay attention to her, effectively, obviously. Essentially, we're going to be going into the next step of our DNA bioinformatics project. So essentially, let's see if I can make this work. Yeah, that doesn't really work that way. All right, so cat, quit it. Essentially, we've got a whole, like a flow chart, right? This is really kind of like the conceptual framework that we're working with in here. So you have your, I don't know, gene of interest, and you want to sequence it to figure out the link between its sequence, whether regulatory or coding, and the phenotype you're observing, right? So essentially, the first thing is obviously defined, right? A phenotype of interest, and B, trying to figure out what the gene of interest may be. And back in the olden days, before super cheap whole genome sequencing was available, this was actually a lot of work, right? So typically what you would do is try and figure out what chromosome it was on first. And you would do that typically by looking at, relying on the principles of independent assortment, right? So if it's on, let's say, chromosome 1, then you would expect to see lower levels of independent assortment. You'd essentially expect to see some degree of linkage, right? Which you would not see with all the other chromosomes, right? So you can narrow it down to that chromosome, and then you would use a series of genetic markers along that chromosome to try and figure out where it was, and you get closer and closer and closer, and then you'd sequence the actual region and try and figure out what was there. Now life is a lot easier for scientists. We just, ship it off and, I don't know, spend, I don't know how much it costs now, maybe $500,000, something like that. And then it will just tell you where the mutation is and in what gene. Right? But anyway, that's kind of, modern, et cetera, et cetera. So typically you need to do, you need to get your cells or tissue of interest. You need to do a DNA extraction because you can't do really much molecular work without getting your nucleic acid materials out of the cell. Part of the reason is because they're in a cell, right? So, they're not readily accessible to whatever you want to do with them. And the other part is that there's a whole bunch of other stuff in the cells which interfere with basically molecular biology. Either inhibitors of polymerases or things that will degrade. Nope, don't want to do that just yet. Things that will degrade your molecules of interest, a bunch of different stuff, right? So essentially you want to get your DNA, if it's a gene of interest, RNA, if it's expression, out of the cells. That's what we're going to be talking about today. Then you need to, oops, can't spell. You need to make sure that the DNA is of sufficient quality, right? That it's not degraded or contaminated with a bunch of other junk. Because remember, again, that other junk, like in bunny ears, can really severely interfere with your downstream processes, right? Again, using that same kind of terminology that we use with gene structure. Once you've done that, make sure it's your DNA is of sufficient quality for, say, PCR or whatever it is you want to do with it. Then you do your, oh no, I'm not going to write that info. Don't think I have enough ink. PCR, right? So PCR basically equals amplification. And I was trying to be smart by writing it big and I ran out of space. Right, so essentially, if you have one cell, let's say, and you extract the DNA from that one cell, in a diploid organism, you're going to have 2 copies of that gene, right? Which is a small amount of not very much. Right? So to be able to do anything with that gene sequence, whatever it is you want to do, cloning, sequencing, putting in something to express it as a protein, you know, for vaccine work or whatever, right? You need a lot more of it, a lot, right? And so you're never going to get enough DNA by just extracting it. You have to amplify your region of interest. And that also has the benefit of kind of narrowing down the focus of whatever you want to do subsequent to that. So for example, as far as I'm aware, the COVID-19 diagnostic at present, it doesn't look for DNA, it actually looks for RNA, which is the presence of replicating viruses rather than viruses that may have been there in the past. But it uses the same principle, right? You're extracting nucleic acid material, you're amplifying it, and then essentially use that amplified product as a diagnostic tool, right? That's why it took a long time to get tests up and running, particularly in the US, but also other places, because you have to figure out this stuff first and make sure it's accurate, right? It's reproducible and you don't have many false positives and very few false negatives, right? So we need to amplify a whole bunch of this material so we have stuff to work with. Now, again, the flip side is we're left with a bunch of junk in there that we want to get rid of. And I'm going to run out of space on this board. So basically, you essentially do another PCR DNA purification. Similar processes to what you're doing up here, but kind of simplified because you don't have to worry about cells and stuff like that because you don't have any cells here. It's just enzymes and salts and things. You've got to get rid of those so that they don't interfere with whatever subsequent step you're doing. So that's the cleanup part. And then obviously you don't want to lose it all, right? Because then you'll be screwed. So you need to just do a quality control check to make sure it's still there. Then once that is all okay, and again, I want to really emphasize that this is very much what you would do in a research lab. I've done this myself a ton of times, finding out, you know, working on different genes and making different things and looking at expression and so on and so forth, right? It's a very fundamental process in any kind of molecular biology, whatever field you happen to be in, right? You could be working on plant stress resistance, or you could be working on human cancer. You know, the principles are the same. So once you've got this all cleaned up in a nice little tube, you send it off for sequencing, typically with primers or little bits of DNA specific to your gene of interest. So remember again, this is your whole genome of that organism, including the gene that you're interested in, but also tens of thousands of others. You clean it up, and then you basically narrow the field down, ideally to just the bit that you're interested in, right? So that when you sequence it with primers, which essentially recognize just that bit of DNA, and we'll talk about primers and what they mean later on when we get to PCR, then you're able to generate sequence information for that gene of interest. Now, this is really cool. It's a really cool process. It's how the human genome sequence was produced and worm genomes and stuff like that. It's a bit different now. Like the technique for doing this is not the same for the one that we would use to sequence a whole genome, because, you know, technology has moved on a lot in the last 20 years or so, like since I've started my career, basically. And now we use a much higher throughput, faster method that's incredibly cheaper. How much did it cost? I think it cost something like, what is it, $1.5–2 billion in today's money to sequence the human genome for the first time. That was in 2001, thereabouts. Do you know how much it costs now? About $1,000. So that just gives you an idea of how much technology has advanced in terms of this technique. We don't use that for like small runs because it's not cost effective. We just pay like $5 a pop, basically, for each one. And then once you get that, you have your bioinformatics, right? And really what we mean by bioinformatics is comparing sequence information in a computer. So we want to make sure that, well, yes, we sequenced the right thing. It wasn't like some like butt protein from pigs. You know, we didn't get some random ass thing, literally, produced. And that does happen. Don't know why. But we also want to then compare what we get for our gene of interest and our mutation, which presumably produces that phenotype. And we want to compare it with what is considered normal, right? That's what we're going to be doing with our bioinformatics section, right? So this is and again, this is really what you would be doing yourself. In fact, when I was a PhD student, I cloned for the first time a particular gene involved in cell signaling from a parasite. And this was exactly the process that I used, right? It's a little bit more complicated because the PCR wasn't as clear cut as the one that you would have been doing, because we didn't know what the gene sequence was. But eventually we ended up getting there. It's a lot of work as well. I think that was probably about, how long did that take me? 6 months, something like that. Yeah, I wish you could too, Angela, but you know, hold on to the hopes that, well, don't want video, that you'll be able to do this in subsequent courses. I really do, really fingers crossed, because it's a cool process to do, and you know, ideally you'll get to do it in some subsequent career, right? Because this isn't the end of your biology career, right? This is not even the beginning of it, to be honest. And trust me, after a while, you know, yeah, it is really at the end of the day. You know, one hopes that once you get through, you know, genetics, right, without issues, and then go on to complete the rest of your degree, that then you'll be able to use that to do something bigger and better. Whether that's working as a, you know, a lab assistant, or doing a master's, or doing a PhD or MD or whatever, you know, it's the beginning, very much. You know, I don't consider, I have not reached the end, you know, of my particular trajectory. Hopefully I'm not too far off it, right? Because I've been doing it for a while, but I certainly am not, I don't consider myself as being, you know, a sage on the stage, having done it all. You know, I'm still, yeah, you know, I think I can see what you mean, right? That you don't want to feel kind of like you're behind. Really what you're missing from not doing this in person, pardon me, is the motor memory, right? The actual pipetting from here to here, here to here, here to here, things like that. So that's what you're missing out on. It is important, but it's anybody can learn how to do stuff, right? That's what I could teach you how to do RNA interference in worms. It would take like a month, maybe 6 weeks of meeting once or twice a week, right? It's just, it's like learning to ride a bicycle, you know, any of that kind of basic motor skills type stuff. But the really, really big difference is that if you don't understand what it is you're doing, right, the principles behind it, the process, the interpretation, then you're only going to get to a certain level, right? And all of that stuff, I'm hoping, right, I'm doing my very best to make it so, is what you'll still get from taking this. So it doesn't matter how well you can do PCR if you don't understand what it is that you're doing. Because even if, you know, even as a lab assistant, right, working in a research lab or a pharmaceutical lab or even a like a testing lab at a hospital, right? If you don't understand what you're doing, you will be forever limited in what you can do, right? You know, so yeah, and everybody has to start somewhere too, right? And so really what you're learning here is not the actual mechanics of doing something. And even if you were to do this in person, that's really not what you're doing because it doesn't take long to learn how to use a pipette. It's like an hour maybe, right? What you're learning is the stuff that goes on up here. That's what really differentiates people who can do a PCR versus the people who can design a PCR, right? It's it's like, you know, it's easy enough to follow a recipe and end up with a nice cake, right? It's very different to actually figure out a cake of your own. I don't know. I like cake analogies because I like cake. But anyway, so that's kind of the idea behind it, and that's what I'm a very, very firm believer in that because I've worked in lots of different fields, I've learned lots of different things, and I've worked with lots of different people. Yeah, baking is a kind of science stroke art. You know, I'm not 100% sure about it just being a science. Yeah, it kind of is. Yeah. Anyway, is that it's the people that are really truly good at stuff aren't the people who can do things. It's the people who can think, who can understand a process and join different things together, right? And so that's really what's valuable to learn, right? In my opinion, because anybody can learn how to do something. It's really none of the things. Yeah, I mean, yeah, even doing like microarray analysis or large scale transcriptomics, anybody can do the mechanics, right? I actually got a student, spent 3 months, who extracted gene IDs and function and location of expression, stuff like that. You know, she just did it in a kind of spare time as a research project. It's not hard to teach her. She wouldn't actually necessarily understand what it all meant, right? Because again, that's a different level, right? That's kind of where my job comes in. But actually teaching us to do it is trivial, you know. And so that's the difference, right? It's like yeah, I don't know, I run out of analogies essentially. But yeah, it's the thought processes, it's the scientific method, right, that matters. Actually doing the work, you know, you'll pick that up whenever you come across it, right? And to be honest, in terms of CV type stuff, in terms of going further on, none of your undergraduate courses really count for much, to be honest. Your ability to do independent research with a professor or internships or volunteering at hospitals, stuff like that, that's what really matters, for getting into grad school, for getting jobs as a lab assistant, stuff like that. Your GPA doesn't hurt, but it's not what will get you a job or a grad student position. It's really independent experience showing that you understand the scientific method, basically. Anyway, tangents for the win. Yeah, and the earlier you get that sorted, the better, to be honest. And it's hard at the moment, like, for example, what Ali just wrote about opportunities shutting down, and that's very real, but it shouldn't stop you from trying, essentially, right? As soon as things go back to a little bit normal, you need to be out there kind of basically just pissing people off until they give you a position. Because those are the people that are going to write you references, and those references are what are going to get you your position, essentially. And references from a professor, they don't hurt, but I haven't actually met any of you, right? But even if I was teaching you in person, I would have known you for 3 months, right? Which isn't and not like one-on-one personal relationship type deal where I work with you for a long period, right? So my ability to actually speak to your ability to do whatever job it is you're applying for is fairly limited. It certainly doesn't hurt if that's all you got, you know, but you don't want it to be all that you've got. This has turned into like career advice lab anyway. So what we're going to be doing today is the DNA extraction part. Man, I was waving my Sharpie around too much. I need to get another one of these. Actually, I'm going into work tomorrow, so. Just have to make sure I don't wash my hands between now and tomorrow morning. All right, let's work on it. Yes, it will. It's very effective. Actually, I have a graded range of pens that I use depending on how important it is to remember something. Permanent markers are the best. Like if I really need to remember something, like having to shed skin to get rid of it is the way to go. So essentially, this is what we're going to be doing today. And there's a assignment, did it this morning, on this in the module 2 course content folder. And really it's going to be, it's a bunch of questions and short answers and stuff like that based on what we're going through now and on the lab report, lab handout, sorry, itself. So it shouldn't be anything in those questions that you don't already know at the end of this or from reading the handout. It's due next Tuesday 14th, I think, midnight. Yeah, so you have over the weekend to do it, or not. Haha, it's your case. Sorry, haha, that was a little bit of a dig. Yeah, it shouldn't take you very long to do. Probably be able to do it this afternoon, I'd expect. And this video will be up probably within about an hour or so of us finishing talking, because it takes a while to convert the video and then upload it to YouTube. So this evening it'll definitely be available then. Anyway, so essentially you start with your cells, right? Whatever that might be tissue from a part of the body, might be whole organisms, you know, whatever it is. Then what you need to do is you need to smash the crap out of them, right? So you need to make sure that you have a kind of like a cell soup, basically, a homogeneous mixture of cells. So if you have, I don't know, let's say muscle tissue, right? It has like, you know, chunk of whatever. You want to make sure that you have broken apart every single cell from the cells that it's next to. This isn't actually breaking the cells open. It will do it a little bit, just mechanically rupturing cells, but it's not very effective. This is all about, let's do it over this way. This way, perhaps. This is really all about making sure that all those cells are available to be ruptured, right? Which, if you imagine, you put a chunk of steak in a tube and you try and lyse those cells, steak cells, right? You're only going to be able to rupture the ones on the periphery, right, around the outside of that piece of tissue. So that's not going to be a very effective way of isolating DNA. You want to make sure that that structure is completely broken down. And this is the homogenization step. Sorry, kind of chicken scratchings. You have to go by what the video says. Right, so this is, you can do it in a bunch of different ways mechanically, right? So you just like put it in a blender, basically. You can get little mini blenders, which are super cool, very, very noisy. You can smack it with a giant hammer, basically. It was called a French press. And so you can stick in a small hole and then just like smash it with a hydraulic ram. That does the trick too. Oh, life's hard, cat. And you can also freeze store it. So what I do with my worms, because they're very, very tough, is I freeze them in liquid nitrogen and then grind them mechanically, right? And do that multiple times, and that then essentially homogenizes everything. That actually also does help lyse them as well. So the next job is you've got to get into those cells or get the DNA out of them, or RNA if that's what you're working on, or even proteins, right? Depends on what it is you're after. We're after DNA, so we want to get the DNA out and basically get rid of all the other stuff, right? This is the lysis step, which means breaking down open. To lyse something is to break it open, right? That's just what it means. And so typically we use stuff which screws around with phospholipid membranes, right? Because phospholipid membranes aren't very strong, but they can hold themselves together pretty effectively. So, I mean, you could even like just add like red blood cells to water, for example. That will cause them all to lyse based on inflow of water due to osmosis. Tried that with plant cells, nothing's going to happen, right? Because they're much more robust. So you need something else. And so what typically this involves is a detergent. Right. So it could even just be something as simple as Dawn. We don't use Dawn in labs because, you know, we're proper scientists and stuff. We use things like a compound called sodium dodecyl sulfate, or SDS, which is properly nasty. And it will just cause cells to pop straight open, basically. Right. So the issue then is that this is a giant mush of phospholipids, storage lipids, RNA, protein associated DNA, a whole bunch of junk that you don't want, right? And so we need to start getting rid of stuff in here before we can even think about getting our DNA out of that mess, right? And so what I'm just going to call it DNA and junk, right? What we do now is what is called RNA protein degradation. Long words is a pain on a very small whiteboard, right? So we want to break, so this mush of lipids and protein and RNA and DNA, we want to basically destroy the RNA and the protein. Sometimes we don't care too much about the RNA, it'll just end up degrading on its own. Certainly the way that we do it doesn't take that into account, but we very much need to get rid of protein. Sorry, I just have to show you an image of my cat. She's being very cute. Look at that. Isn't she adorable? Right, so yeah, you know, a hard life, lying around doing absolutely sod all. Question. What is, if you want to get clean, pure DNA out, right? Nothing but the DNA. What protein above all do you need to get rid of, particularly if we're talking about eukaryotes? Here's a clue. There's a particular group of proteins really important in eukaryotic genome organization. Need another clue? They form a complex with DNA, and that complex is called chromatin. Come on, you got to remember what the protein that is bound to DNA is called. Begins with an H. Ends with histones. Almost, that was a good guess. Histones, right? So helicase would be the case if that DNA is replicating, which it may be, but isn't going to always be the case for whatever cells you're isolating, right? So if we want to get good, clean DNA out, we've got to degrade protein. Also, another extra benefit of that is amongst these proteins are things that will actually degrade our DNA, right? So we also, or whatever, or interfere with whatever else we want to do afterwards. So we have to get rid of this stuff, right? And end up with DNA and junk. Now, obviously, we don't want no junk. We want just DNA. And so the next step, and this is my, it's kind of like simplest part, but also my favorite part in doing DNA extractions. Hang on, let's think of a good way of writing this. Is we put that on a little column called a spin column. Let's see. Go away. So essentially, let's see if I can make these a little bit bigger. Let's make that a bit bigger. Ah, come on. Sorry, someone was saying something in chat, I think, but I can't see what they're saying. Anything important?
Yeah, it was just me. I was asking if on the whiteboard that says DNA band or DNA bound.
Bound. I'll show you why in a second. And so, like the old school way of doing this would be to precipitate the DNA out of the solution, right? So essentially you would add, typically isopropanol is the alcohol of choice, which is, DNA is not soluble in, because DNA is 100% polar and alcohol is only a little bit polar. And so if you add isopropanol to your DNA and your junk, then, and that's in the non-American terminology, then essentially your DNA will precipitate out and then you can physically remove it, right? And sometimes you can still do DNA purification like that. What we would do and what is much neater and easier is to bind that DNA to something. So essentially everything else then leaves, right? And that's what these spin columns are, right? So if you look here, right? So typically you have outer collection tube, and then you have a smaller tube that goes in that. If you look really closely, actually, this one's a better picture. You see that white solid bit at the bottom of the column? It's actually pretty clear in this image at least. Well, that is a silica mesh, like filter, essentially. It's like the foam in the end of a cigarette, kind of, right? And at the pH of the binding solution, the solution that the DNA is in and all the other junk, right? DNA and only DNA will bind to that silica mesh. And so if you spin this, you put your DNA and junk at the top here, and you spin it, essentially centripetal force will result in all of the liquid going through that filter, right, until it ends up in the collection tube at the bottom. But the DNA will stay in that white silica mesh. So essentially what you end up with is all your junk at the bottom, your proteins and lipids and RNA fragments and, well, whatever, right? And relatively clean DNA at the top, still in that tube. So that's what I mean by bound DNA, because that DNA is actually tightly, very tightly bound to that mesh. And you can see that here, essentially it's a silicate substrate. And along with some ions, also called chaotropic salts, allows DNA to bind to that silica, right? And it is very tightly bound. You don't tend to lose very much of it. Does that make sense? I can't see. Where are people? Chat's not coming up again. So this is a situation that you can manipulate, right? Depending on the pH of the solution that goes over it. So let's stop that. Oh, here you go. This is a little bit convoluted. This is where I wish I had a little bit more space, right? So essentially that's bound DNA plus salts. So essentially the things that are part of that binding solution, right, that this is here, that also includes some salts, and those will also bind to that silica. Hang on, let's add a little bit of info here. So that's our spin column, allows us to bind our DNA and a few salts and stuff. And all the other stuff, the junk, essentially, is just thrown away, tipped down the sink, unless you want to use it for something else. So we need to get rid of those salts. And we do so with a DNA wash. So we just wash some usually ethanol containing wash buffer over that DNA, and that gets rid of as much of that salt as possible. Doesn't get rid of all of it, but it gets rid of most of it. And because that DNA is still very tightly bound to that silica, and we haven't changed the pH, the DNA stays behind and all the other stuff gets washed off. Okay, again, if you've got any questions, just stick, well, metaphorically, stick your hand up. And then this isn't going to do us no good if it's stuck to a bit of plastic inside a tube, right? So we need to get this out of here. And hey, managed to do it. I'm going to have to write this a little more carefully. Now what we need to do is elute the DNA, right? Which means to unstick it essentially from our mini column. And we do that by, actually it's a neutral pH. So if we can use either ultra pure water, right, which has been distilled and purified and filtered and stuff a lot, or we can use another solution which reduces nuclease activity called TE. And it says basically we just run the liquid over the silica mesh, and because of the change of pH, that means that DNA isn't bound anymore, right? That changes the ionic state essentially of the, I'm not sure if it's of the DNA or the silica, I don't actually know. But either way, that DNA then goes into suspension, into the solution, and it ends up at the bottom of that collecting tube. And then you have your DNA. Well, actually, then you put it on ice before it degrades, and then you have your DNA. You stick it in the freezer or in the fridge. And so that's basically the process, right? We need to homogenize the cells so that we have a homogeneous cell soup, because we want to lyse every single cell there to get the most DNA possible. We don't want to just do the stuff on the outside. We then lyse those cells, so we break open those cells, and it's essentially a giant mush of cell membrane fragments, proteins, DNA, organelles, all kinds of stuff, right? It's a messy mess. And then we want to, at the very least, get rid of any proteins, or at least not, if not get rid of them, then break them down as small as possible, right? And that's especially important for eukaryotic DNA, because we don't want DNA bound with histones, right? We don't want chromatin, we want DNA, right? And so at the very least, we have to degrade the proteins to get rid of that, right? And so that then leaves us with, again, a more refined mush, basically, right? It's our intact DNA, one hopes, and this is what the DNA QC will tell us later on, and a whole bunch of stuff that we don't want. And the quickest, easiest way of getting rid of that, well, A, you could either take the DNA out of it, which is what alcohol precipitation would do, or you could keep this behind and then get rid of the junk. And that's what we would do with a spin column, right? So we get our sample, we put it in the top of the spin column, we spin it really fast, and then all of that junk, literally, flows through the column into the collection tube and is discarded. The DNA sticks or is bound to the column, to that silica in the column, by ionic interactions with the help of some salts, right? And so then the last steps are really just cleaning up that DNA to get rid of any of those salts, because salts do interfere with, like, PCR, for example, in a big way. And then once we've cleaned it via a series of washes, then we change the pH of the solution that is going through, and the DNA simply floats off and then gets spun out into the collection tube at the bottom. That's how you do DNA extraction. Ta-da! That is a crazy, messy whiteboard. Hopefully it makes some sense. Especially with my not completely legible handwriting. Okay, so again, obviously you can't do this yourselves. Actually, you can do a DNA extraction yourself if you want. Just get some rubbing alcohol, some Dawn, and some tomatoes, or strawberries, bananas, and you can do your own DNA extraction. It's quite a lot of fun actually, and the principles are exactly the same. So those kind of high school Gen Bio 1 DNA extractions from, I don't know, yeah, it's pretty fun. Using just basic household ingredients. Actually, meat tenderizer is the protein degrading part. You do exactly the same stuff. You know, you're going to be smashing up strawberries, the homogenization, add some Dawn, which is the lysis step, meat tenderizer for protein degradation. I don't actually know how that works, but it does. And then get the DNA out by doing an isopropanol precipitation. And then you literally fish it out with a, I don't know, a chopstick or something. So you're following exactly the same principles. The DNA is not going to be as clean because you're going to be less able to get rid of the other junk and the salts and the Dawn and the other meat tenderizer bits and stuff they would use. So that DNA wouldn't be as good for further processes like PCR. It would still be usable, but it wouldn't be as good as doing it this way. Because this way you get a little bit more control over how clean that DNA is. But the principles are exactly the same. So yeah, look up kitchen DNA extraction with kitchen ingredients or something, and you'll find that's actually a really fun thing to do with the kids. Should probably do it with mine. Not going to use strawberries though, because I like strawberries too much. Maybe lentils. It is. It is. It's a lot of fun. It's very hands-on as well. Yeah, maybe not playing around with dish soap and meat tenderizer if they're under the age of like 3 or 4, maybe even 5 or 6. Yeah, 10 months. Yeah, you should be feeding him the strawberries and the bananas, you know, not mashing them up, or actually mashing them up and then feeding them. Anyway, that's pretty much it for this. So this will go up on Blackboard along with the video on YouTube, which will be as quickly as I can after this recording gets converted. And then really the assignment is just answering questions which I've pretty much already answered for you here. If you have any particular like issues or whatever, then just send me an email and I'll answer them as best as I can. But it shouldn't take very long, and it's supposed to be fairly straightforward. Just to make sure that you paid attention basically to this and read the lab handout. And that's it. Has anybody got any questions? No? Oh, I'm getting a lick from my kitty cat. She actually, I came in from the garage, I was covered in like grease and sweat and stuff. She loved it. Licked my whole hand clean. It's obviously got no sense of taste. All right, cool. Okay, so have a fabulous weekend, everybody. Stay safe, and I'll see you on Tuesday. Ta-ta for now.